Good afternoon. My name is Suparna Wedam, and I'm a breast medical oncologist in the Office of Oncologic Diseases at the FDA. I would like to thank the organizers of the San Antonio Breast Cancer Symposium for inviting me to speak in this educational session regarding emerging adjuvant therapies. Shown here is an outline of the topics I will cover today. I will start with some regulatory background. Then I will review clinical trial endpoints that have been used in the early breast cancer setting, followed by a few case examples highlighting how these endpoints have been used in different trial designs. Any drug approved by the FDA should have substantial evidence to show that it is safe and effective. There needs to be sufficient information to determine that the drug is safe for the use under the conditions recommended in the proposed labeling. Substantial evidence of effectiveness is based on adequate and well-controlled investigations to support that the drug will have the effect described in the proposed labeling. An overall survival improvement is not required for approval. However, the benefit-risk assessment must be favorable. Now, while the FDA has a lot of responsibilities, there are also some misconceptions about the scope of our regulatory authority. The FDA does not regulate the practice of medicine. We also do not take into account payment issues. We know this is a very important problem. However, it is not under our purview to consider this in decision-making. There are two approval pathways available in the United States: regular approval and accelerated approval. For traditional or regular approval, a product must demonstrate direct clinical benefit, meaning that there is an improvement in survival or symptoms. The therapy must be more effective than placebo, but there is no comparative efficacy requirement for regular approval. Accelerated approval was introduced in 1992 based on the need to expedite drug development for serious or life-threatening illnesses. The product should provide a meaningful therapeutic benefit over available therapy, and approval is based on an early endpoint reasonably likely to predict clinical benefit. For products granted accelerated approval, there is often a requirement to conduct post-approval trials to verify the anticipated clinical benefit. The same statutory standards for safety and effectiveness must be met for either approval pathway. It is not surprising that the accelerated approval pathway is used often in oncology, since all the diseases we treat are considered serious or life-threatening. This pathway comes with some advantages and challenges. Advantages of this pathway include permitting the use of an earlier endpoint. An early endpoint, in turn, usually means events occur earlier so that trials can be designed with a smaller sample size. Ultimately, this allows for marketing promising new therapies sooner so that they may be available to the patients that need them. However, there are some challenges. The product must demonstrate that it is better than available therapies. In this setting, available therapy refers to any therapy that has received regular approval in the United States for the same indication being considered for the new drug. This is not required for regular approval. And due to the uncertainty of using an earlier endpoint, post-marketing trials may be required to verify meaningful clinical benefit. Accelerated approval indications may be withdrawn if post-marketing trials do not confirm clinical benefit or are not conducted with due diligence. Accelerated approvals are meant to serve patients. If post-marketing clinical trial data do not demonstrate clinical benefit, or there is a lack of trials conducted to confirm benefit, and alternative therapies exist, patients may not be served by continuation of the original accelerated approval. Now, moving on to clinical trial endpoints. Direct measures of clinical benefit are based on prolongation of life or a better life. In other words, they measure how a patient feels, functions, or survives. Some examples of this include overall survival and patient-reported outcomes. On the other hand, early endpoints used in the accelerated approval pathway are reasonably likely to predict clinical benefit An example of this used in early breast cancer is pathologic complete response. This diagram shows different endpoints that may be utilized along the course of treatment for early breast cancer, starting with the earliest endpoint on the left, moving to the right. In the setting of neoadjuvant therapy, followed by surgery, pathologic complete response, or PCR, is the first endpoint that may be considered. The FDA-preferred definition of PCR is the absence of invasive breast cancer in the breast and axilla. These same patients may then be followed longer to evaluate for event-free survival. This terminology is used for neoadjuvant trials in which all patients, by definition, will have invasive cancer at the time of randomization. EFS is defined as time from randomization to any of the following: progression of disease that precludes surgery, disease recurrence, or death due to any cause. Whereas when patients are treated in adjuvant trials, there is no invasive cancer at the time of randomization. Therefore, the endpoint to use in that setting is either disease-free survival or invasive disease-free survival, defined as time from randomization until recurrence or death due to any cause. Now, definitions for DFS and IDFS often overlap and have varied from trial to trial. With continued follow-up, overall survival is the last endpoint that may be used in either the neoadjuvant or adjuvant setting. This is defined as the time from randomization until death from any cause. Each endpoint has strengths and limitations. Overall survival is often considered the gold standard. It clearly has clinical benefit with no bias and accounts for both safety and efficacy. However, this endpoint is not always feasible and takes a long time to assess, especially in an early stage setting where it is common for patients to have long-term survival. In addition, the results may be confounded by subsequent therapies or crossover if allowed on study. Non-cancer deaths are included in the definition, which may dilute the efficacy effect. IDFS, DFS, and EFS allow for smaller size trials of shorter duration compared to overall survival. Randomization is still required for these trials, just as with overall survival, as time to event endpoints cannot be accurately interpreted in single-arm studies. These endpoints are not affected by crossover, and the progression that is captured as part of these endpoints is usually considered clinically relevant, which is why these endpoints may be used for regular approval. However, use of these endpoints are potentially subject to assessment bias, especially in open-label studies where the investigator and patient are both aware of which treatment the patient is receiving. Timing of assessments must be the same between arms. Since imaging is not done routinely in early breast cancer, this means that clinical evaluations assessing for progression of disease should be done at the same time. Again, non-cancer deaths are included in the definition. Finally, pathologic complete response allows for earlier assessment and a smaller sample size. A trained pathologist blinded to treatment assignment is required to evaluate the surgical specimens for this endpoint. There's ample evidence showing a correlation between PCR and survival at an individual patient level, but not with long-term clinical benefit at a trial level. Due to that uncertainty, this endpoint may only be used for accelerated approval and requires further confirmation of clinical benefit. This table is an overview of the drugs approved in the early breast cancer setting over the last decade. Only one was granted accelerated approval based on an early endpoint of pathologic complete response. The other 5 were granted regular approval based on accepted endpoints of invasive disease-free survival or event-free survival. This includes the regular approval for pertuzumab, which was converted from the accelerated approval after confirmation of clinical benefit. Next, some clinical trial designs and endpoints based on case examples. One of the biggest changes in early breast cancer clinical trial designs over the last decade has been the incorporation of pathologic complete response as an endpoint. To learn about how this endpoint could support approval, the FDA established an international working group known as the Collaborative Trials in Neoadjuvant Breast Cancer. Following wide discussion at public workshops, a guidance to industry regarding the use of PCR as an endpoint to support accelerated approval was finalized in October of 2014. The guidance outlined potential development strategies, including single and multiple trial models, and appropriate high-risk patient populations that could be considered for this approach. With advances in treatment, particularly in the setting of residual disease following preoperative therapy, the guidance was updated and finalized in July of 2020. In addition, we published our perspective on how to leverage the PCR endpoint with the evolving treatment landscape. The following case examples highlight these various approaches. In the first example, 417 patients with HER2-positive early breast cancer were randomized to one of 4 treatment arms prior to surgery. The primary endpoint was pathologic complete response. Further systemic therapy was administered in the adjuvant setting. Outlined in the red box are the 2 arms of interest, which allowed for isolation of the pertuzumab effect. The improvement of pathologic complete response observed with the addition of pertuzumab was supportive of an accelerated approval. This study was not adequately designed to confirm long-term clinical outcome such as EFS or OS, and therefore, this is an example of a multiple trial model in which long-term clinical benefit needed to be confirmed in a separate study. The confirmatory trial had a conventional adjuvant study design in which 4,804 patients with HER2-positive early breast cancer were randomized to 2 treatment arms that allowed for the isolation of pertuzumab effect in the adjuvant setting after surgery. The primary endpoint was invasive disease-free survival, with additional efficacy endpoints including overall survival and disease-free survival. The improvement in IDFS was supportive of conversion to regular approval. The submitted application for accelerated approval demonstrated a significant absolute improvement in pathologic complete response rate with an acceptable safety profile and no new safety signals. This application was supported by a statistically and clinically significant effect on overall survival in a separate trial in the metastatic setting, an extensive body of safety data from treatment of patients with metastatic breast cancer, and an adjuvant confirmatory trial that was fully accrued. This application was discussed at an Oncologic Drugs Advisory Committee, or ODAC meeting in September of 2013. Recommendation from the committee was a favorable benefit-risk assessment, and the FDA went on to grant accelerated approval for pertuzumab in the neoadjuvant setting. The adjuvant confirmatory trial subsequently met its primary endpoint of invasive disease-free survival, which allowed for regular approval in both the neoadjuvant and adjuvant settings. This multiple trial model permits widespread access to highly effective drugs earlier because fewer patients need to be accrued before analyzing the PCR endpoint, as seen with the smaller first trial in this example. In addition, demonstrating the efficacy of a drug in multiple trials provides greater assurance that the results are not caused by chance alone. Also, there is less concern about confounding results from the post-neoadjuvant treatment since PCR and long-term clinical benefit are being assessed in separate trials. Finally, this model may facilitate study of the new drug in a different combination or patient population, such as an earlier line of therapy in the confirmatory trial. The principal disadvantage with this approach is the feasibility involved in designing and conducting a second trial. This was not an issue in this example, as the confirmatory trial was fully accrued. However, it is understandable that confirmatory trials that repeat the design in patient populations enrolled in the trial that supported approval would be expected to accrue poorly in the United States, especially once the drug is available to patients. To mitigate this issue, we recommend the confirmatory trial to be well underway, if not fully enrolled, at the time of accelerated approval. And status of the confirmatory trial is important in our benefit risk assessment. In the second example, 1,174 patients with early-stage triple-negative breast cancer were randomized to one of two treatment arms in the neoadjuvant setting, with or without pembrolizumab, followed by continuation of either pembrolizumab or placebo in the adjuvant setting. Co-primary endpoints were pathologic complete response and event-free survival, with a key secondary endpoint of overall survival. This is an example of a one-trial model for neoadjuvant therapy, in which an early endpoint of PCR is assessed at the time of surgery, and within the same trial, the same patients are then followed for longer-term clinical endpoints of EFS and OS. However, due to the differences in adjuvant therapy between the two treatment arms, it is challenging to interpret how PCR may predict for EFS in this particular study. With this example, at the interim analysis, a small improvement in the early endpoint of PCR rate was noted. EFS and OS data were immature, and an increased toxicity, including a small increase in deaths due to adverse events, was observed with the addition of pembrolizumab. This is an especially significant consideration in an early-stage breast cancer population. This application was discussed at an ODAC in April of this year. Based on the benefit risk assessment, the ODAC committee recommended to await further data before approval. Accelerated approval was not granted at that time. At the following pre-specified analysis, there was a statistically significant improvement in event-free survival, which was considered clinically meaningful. Overall survival was immature, but with no detriment. And based on these results, regular approval was granted. With this model, a single trial powered for both PCR and EFS or OS does require a larger sample size than a trial powered only for the PCR endpoint. A single trial has the potential for a drug to receive accelerated approval based on an early endpoint of PCR and then confirmation with long-term clinical benefit within the same trial. The statistical analysis plan for such an approach must adequately account for both endpoints. Because all these patients need to be enrolled before any efficacy analyses, including analysis of PCR, are performed, this approach provides a larger body of safety data from an early-stage patient population at the time of potential accelerated approval. The single trial model also avoids the feasibility issues of conducting a separate confirmatory trial. Disadvantages of the single trial model include the potential to expose a large number of patients to less effective and/or more toxic therapy. Again, this is because the sample size is larger and all patients must be enrolled before any efficacy analyses are performed. Due to these same reasons, there could be a longer wait to initial U.S. approval compared with the multiple trial model. Finally, there is the potential for confounding by post-operative systemic therapy, as seen with this example. When there is a difference in adjuvant therapies between two arms, it is difficult to interpret how PCR may be predicting for long-term survival endpoints. In the last example, almost 1,500 patients with HER2-positive early breast cancer that received preoperative therapy followed by surgery with evidence of residual invasive tumor on pathologic review, or in other words, did not have a pathologic complete response, were randomized to trastuzumab or T-DM1 therapy. The primary endpoint was invasive disease-free survival with additional efficacy endpoints of DFS and OS. An improvement in IDFS supported regular approval in this setting. This trial design enriched for a high-risk patient population, utilizing the space of residual disease after neoadjuvant therapy. Patients who have residual invasive cancer after receiving preoperative systemic therapy are expected to have an increased event rate in trials, and therefore trials evaluating agents in this population are likely to complete accrual and provide results more rapidly than conventional adjuvant trials. No new safety signals were observed, and the primary efficacy endpoint of IDFS was an acceptable regulatory endpoint to support regular approval. In summary, there are two different approval pathways, each with its own advantages and challenges. Although accelerated approval is used often in oncology, it is not appropriate in all cases. It has only been used once in early breast cancer over the last decade. Various endpoints are used in clinical trials studying the treatments of early breast cancer. The use of pathologic complete response has become more prominent over the last decade, both as a primary endpoint and as a selection criterion. In the future, other endpoints such as circulating tumor DNA might be used. However, adequate data need to be available before any new endpoint can be used to support a regulatory decision and should be discussed with the FDA in advance. Endpoint selection depends on the trial design and regulatory pathway. There is no single best development strategy. A benefit-risk assessment is integral for the approval of each drug and must be considered favorable. Finally, it is essential that clinical trial populations reflect the general population to be treated post-approval. Therefore, trials should enroll study participants who reflect the demographics of clinically relevant populations with regard to factors such as race, ethnicity, age, and sex. Listed here are some references along with links for information discussed in my presentation. Thank you for your attention.